but turns out we still don't understand HDL, and we'll talk about it a little bit. LDL, we understand LDL very well, and we'll go after it quite aggressively with LDL lowering therapies. Triglycerides are important and certainly take their share at the risk pie chart. Total cholesterol, and the newcomer in town, lipoprotein little a. It's not much talked about. It's still not in the guidelines to measure it. I think everybody should know their lipoprotein A levels. If you're below 50 milligrams per deciliter, you're safe. But as you see later during the talk, if you're above 50 milligrams per deciliter, that's when the risk, it starts contributing to CVD risk, and it's an independent risk factor. And one could argue that it's worst of them all. So where is this data coming from? It builds from the historic, why can't I advance my slides? Okay, clicking works. It comes from the historic epidemiologic data, such as Framingham presented here, Tromsø, ProCam, that all taught us over the years that high LDL levels are bad, and we are addressing it, but they're only bad when HDL levels are high. That was the belief. When HDL levels are low, that was the belief. If HDL levels are high, high LDL levels might not be that detrimental. That thinking has shifted in the recent 10 years, and we'll talk about that. So when we think about LDL lowering, we are addressing it with aggressive LDL lowering therapies. And here is a result from the Improved study that even when we take LDL cholesterol levels as low as 50 milligrams per deciliter, we are still left with anywhere, depending on the cohort and ethnicity, 30 to 40% residual risk. And our job in the lab is really to go after what defines that residual risk. My dream goal is to modify that risk prediction algorithm to include new parameters and to come up with a better algorithm. So that's my overall goal. And to do that, we need to find the determinants of residual risk. It could be due to triglycerides. It could be to lipoprotein A. Certainly, we'll talk about it. HDL, as I said, is a big mess. We don't understand it. And I'll present how I understand HDL. And certainly there are other risk factors. If we think about residual, the CVD risk as a big pizza chai part, all of these factors take a slice at it. And the more slices we can identify, the more we can chip off the CVD risk. So the lipoproteins are defined by their density, and HDL being the densest of them all, and it's called high density lipoprotein. Then comes the low density lipoprotein, LDL. Lp(a) is as dense as LDL, but it's very difficult to isolate it because it coincides with it coincides with HDL density range. Sorry, I misspoke. Lpa is the same size as LDL. So doing an ultracentrifugation gradient to get Lp(a) out of plasma is tricky, and we have a good method for that we are putting out that is applicable to clinical studies that require low volume. So that was a big milestone in the field, and I think we've done that. And Paul has worked on it. Paul Mueller, who's going to talk next, has worked on it. He's not going to present that data, but stay tuned if you care about how to isolate lipoproteins per density gradients. So when we say, when we look at their sizes, they are from 7 to 10 nanometer all the way to 40 to 50 nanometer in range for LDL and IDL. So I really want to put out that HDL is really, really small. When you think about HDL having less than 10 nanometer diameter in size, so a cell nucleus, for example, is about 1 to 10 micrometer in size. We don't think about lipoproteins in that sense. But look at there, if you want to have a correlation in mind between the sizes, LDL and HDL would be a basketball to tennis ball if you want to have a size reference. And HDL is small. And I think that becomes very important with regard to HDL biology, because it's small, it can go everywhere. It's in lymphatic tissue, it's in spinal fluid, it goes interstitial space, and it packs a proteome that has been identified by the Heineke group at University of Washington more than 10 years ago. And the proteome that HDL packs has been shown to play a role in lipid metabolism. Yes, that's, I think, a given, but also in complement regulation, acute phase response, proteinase inhibition, that all define how HDL particle is going to function. And the name of the game, I think, in HDL field right now is to identify how these proteins come together on an HDL particle. Don't think about these 100 proteins hanging out on one HDL particle. It will make it way bigger than 10 nanometer in diameter. So there is ApoA1 for sure, a cholesterol ester and free cholesterol core, and then how these proteins come together on every single HDL particle defines how efficient HDL particle is at its function. And the most known function of HDL that is well defined is reverse cholesterol transport. HDL goes to the periphery, collects cholesterol, and brings it back to the liver. That is an atheroprotective function. And when we think about the intimal space, where the macrophages, the cholesterol efflux for macrophages matters, ABCA1 is the main transporter, and it's been well characterized to play the important role of driving cholesterol out of the macrophages. So if we think that HDL as a particle, the function of it can vary and is important, then one can think that this reverse cholesterol transport efficiency from particle to particle and from person to person can vary and can have an effect with regard to CVD risk disposition. And Rader group in 2 back-to-back New England Journal Medicine papers have shown using a proxy cellular-based assay that measures HDL particle sterol efflux capacity, and they have shown that sterol efflux capacity, when adjusted for all the other common risk factors, associates with prevalent CAD status better than HDL cholesterol, better than LDL cholesterol. And they've shown this incident CAD status behaves the same way. So suddenly the HDL field started to shift focus from quantity, which is the HDL cholesterol levels, what gets measured in the clinic, to quality, how well that HDL particle is behaving. So, and that is very difficult to measure because, as you have seen, HDL proteome is diverse. There are multiple proteins that play a role in lipid metabolism that are certainly drivers of cholesterol efflux, but there are also a slew of proteins that associate with complement regulation, immune response, proteinase inhibition, which can affect sterol efflux, but do affect macrophages' immune response that in turn can affect how macrophages regulate their sterol content. So that's why I think in part HDL story is not linear and it's complicated and it's difficult to address in the clinic. So when the Rader papers came back to back in New England Journal of Medicine, I tried to understand how sterol efflux works. So here are all the macrophage sterol exchange players that we know, and in the Rader assay, which is a biomarker assay, we need to think about it as a biomarker assay, cell-based biomarker assay. They heavily focused on the ABCA1 mediated pathway. And when we look at that assay more closely, what has been done is that they take EDTA plasma and they treat it with polyethyl glycol to precipitate ApoB containing particles. ApoB containing particles are VLDL, LDL, LP(a), and what is left in the supernatant contains HDL. Yes, it contains HDL, but it contains all the 8,000 other serum proteins. So I wondered whether the associations they were seeing with regard to sterol efflux were really driven by HDL or the other components that could be present in serum HDL fraction. So we did two things. We took macrophages, which is J774 macrophages. And even though they are stimulated with cyclic AMP, we don't know what cyclic AMP does to those cells. Yes, they do upregulate their ABCA1, but we don't know what else gets upregulated because we don't look for it. And macrophages, J774s, have SRB1 and ABCG1, all known contributors to sterol efflux. And SRB1 is a double mediator. It takes cholesterol in, it puts cholesterol out. And Daisy Shaul later in the series will talk to you probably quite a bit about SRB1. And we used another cell line, which is a BHK, baby hamster kidney cell line that has been transfected with human ABCA1. Jack Oram back at University of Washington developed the cell line, and we've been using it because it's very clean. The cells are treated with mifepristone. They upregulate their ABCA1, and we know that there are no other transporters present on the cell. And the assay, most of you do this assay in your lab. Most of you know how this works. And this slide goes for the other crowd that is not familiar with the assay. So the cells are loaded with tritiated cholesterol, and that's what gets traced. The ABCA1 transporter is upregulated, whether with cyclic AMP or mifepristone stimulation. The cells are treated with the acceptor, whether it's serum HDL, which is PEG precipitated ApoB particle precipitated HDL, or UC ultracentrifugation isolated HDL, or whole serum, as you'll see later in the talk. And the tritiated cholesterol is measured in the media and divided by the total cholesterol content of the cells. And the Rader group in 2010 ATVB paper has shown that in J774s, when they stimulate the cells with cyclic AMP, the ABCA1 stimulated mediated compartment gets upregulated. But as you can appreciate from the slide, that the uninhibited efflux get upregulated as well with cyclic AMP stimulation. And I don't want to get too much into detail, but in those big associative studies, that got published in New England Journal Medicine paper, they did associative studies with both components, uninhibited efflux and ABCA1 mediated efflux. So because I wanted to understand what the serum other than HDL contributes to sterol efflux, we went to the mice, the ApoA1 knockout mice, which loses about 80 to 90% of their HDL. And no matter how we do the assay, we've seen quite a bit of efflux in ApoA1 HDL lacking plasma serum. And when we do size exclusion chromatography to understand where the activity is coming from, HDL range fractions in ApoA1 knockout mice are decreased, but where all the serum proteins elute in the later fractions, the non-HDL fractions have a very strong HDL-like sterol efflux activity, both in ApoA1 and wild-type mice. So the next 4 years was after going what defines that second peak, where the activity is coming from. And the way we approached it, we took the size exclusion chromatography and we followed it with the basic steps of protein purification, where these active fractions were subjected to further separation by charge through cation and anion chromatography. And when we did that, both in wild type and ApoA1 knockout mice, we took the active fractions, efflux active fractions coming from both ion chromatographies, and we subjected them to proteomics, and with the hope that some protein will track with sterol efflux activity. And we went through this exercise with wild type and ApoA1 knockout mice. This is an unbiased orthogonal approach. We didn't know what we were going to get. And this is a dendrogram that displays everything that gets associated with sterol efflux. And whether we did cation or anion column, plasminogen was associated with sterol efflux. So that was a surprise because plasminogen is a non-apoprotein and it plays a role in fibrinolytic cascade. It's a zymogen to plasmin, it gets cleaved to plasmin and plays a role in clot dissolution. This was a surprise to us. And as you see, there are other proteins that get associated in both separations. So there are probably also targets and they are free for grabs. So if anyone is interested, you can go after them if you want. But we went after plasminogen, and plasminogen is a Kringle protein, and we'll talk more about it towards the end of the talk. And it's an evolutionarily spliced variant of LP little a. Do you see how I run in full circles? We're going to come back to LP little a. We're going to talk about it. Okay. And long story short, plasminogen elicits ABCA1 mediated sterol efflux in a concentration and time dependent manner. And because this was a cell line, we also went to peritoneal macrophages. We observed the same thing. We went to bone marrow derived macrophages from ABCA1 heterozygote and ABCA1 null mice to show that plasminogen mediated efflux really depend on the presence of ABCA1. And then because plasminogen gets cleaved to plasmin, the next question was whether plasmin can elicit sterol efflux. And plasminogen gets cleaved to plasmin by UPA, and as you can see here, as we cleave plasminogen to plasmin, the sterol efflux activity decreases. So our next questions are, does plasminogen bind lipids? Plasminogen is the largest pool of oxidized phospholipids in plasma. It's a sink for oxidized phospholipids. So we know it binds lipids, but does it bind cholesterol and how things come together? So today, due to time limitation, we're not going to talk about that, but maybe you might take my word of it. If we do molar comparisons with ApoA1, which has a strong cholesterol and phospholipid affinity, plasminogen and ApoA1 behaves almost the same. So that's very interesting. And I don't know what it means when you think about plaque physiology. So our next question is, does plasminogen bind ABCA1? Plasminogen is a very tricky molecule, very cringly, hard to work with. Not only that, but it has 13 identified cellular receptors. So one could envision that everything we see with regard to cellular efflux could be requiring ABCA1, yes, but ABCA1 could be chaperoning a plasminogen receptor X interaction. So this is what we're going to talk about next. So that was very difficult to address. First, we went to design flow cytometry assay where we labeled ABCA1 with GFP and mixed it with non-ABCA1 expressing cells. And you can see that it's mixed almost half and half here. And then treated cells with plasminogen and stained plasminogen, and then monitored double positive cells. And as you can see, the double positive quadrant increases, but so does this quadrant where the plasminogen binds to its own receptors on the cells. And when we look at the cells as they go through the flow cytometer, this is an image stream technology, which is super cool. Here is the light image of the cell going through the cytometer. We have the GFP stained ABCA1. We see plasminogen. And when we overlay the two signals, when you look closely, you can see where plasminogen binds to its own receptors and where plasminogen co-localizes with ABCA1. This still doesn't address that plasminogen binds to ABCA1. It still leaves the possibility that plasminogen interacts with its own receptor. That's where ABCA1 sits in closer proximity, chaperoning the whole thing. We did co-IP experiments to show that ABCA1 pull downs were positive for plasminogen. And then we went to bilayer interferometry experiments where the sensor is coated with plasminogen, gets dipped in cells that express ABCA1 and don't express ABCA1. We see a nice binding curve when the sensor is dipped in ABCA1 expressing cells. Same with ApoA1, and BSA was our positive control. So we gathered enough evidence to get a small grant that allowed us to do some modeling between ABCA1 and plasminogen interaction. And Shanti Nagarayan, who is a computational chemist, started modeling how ABCA1 and plasminogen could be interacting. The grant, we could apply for the grant because the ABCA1 structure just had got published now over a year ago. And we took advantage of that. And she comes running to my room, my office one day saying, "Natalie, they fit like a glove." This was unexpected. I still didn't think that we would get a hit, but apparently the Kringle 4 domain of plasminogen fits like a glove to ECD two domain of ABCA1, still leaving a hydrophobic opening to let the cholesterol flow through. So this was very encouraging, and we took the high energy hits and we started mutating ABCA1. And we have still in the middle of it. I am fortunate, we are very close, I think, to identify ABCA1 plasminogen interaction sites. Unfortunately, I'm not ready to expose them yet, but as proof of principle, what we did first, we took the very well-known ApoA1 ABCA1 interaction site, which is C1477R mutation. We did that as a proof of principle to show that once we mutate ApoA1 interaction site, which according to the modeling distinct from plasminogen ABCA1 interaction sites, we will still maintain plasminogen activity. And to measure activity, we monitor STAT3 phosphorylation and sterol efflux. So first we made sure that the mutated ABCA1 moves to the surface and it's expressed. And we measured sterol efflux, and like we predicted, when the cells are treated with ApoA1, sterol efflux goes down because that's the ApoA1 interaction site. We successfully abolished that site. But plasminogen maintains sterol efflux when that site is mutated. So that goes to validate the in silico modeling that the sites identified for plasminogen are distinct from the sites of ApoA1, ABCA1 interaction. And when we monitored STAT3 phosphorylation, we observed the same pattern in the wild type cells. Both ApoA1 and plasminogen stimulate STAT3 phosphorylation. And in C2R mutated cells, while ApoA1 mediated STAT3 phosphorylation is abolished, plasminogen mediated STAT3 phosphorylation is maintained. So we have, I think we have one good hit. We need to finalize some experiments before I can come out and say that we have a plasminogen ABCA1 interaction site identified. So stay tuned. So now I can confidently add to this picture plasminogen as an ABCA1 mediated sterol acceptor. So when we think about the whole picture, at the end of the day, what does the plaque see? The plaque doesn't see isolated HDL. The plaque doesn't see just plasminogen, and we know nobody pays attention, but LDL is a double-edged sword. LDL can put cholesterol in, which is the first step in pathogenesis of atherosclerosis, but LDL is also a sterol acceptor. And when we think about plaque development, we need to take into account all the players that come together in total serum. So we went and took plasma from 7 individuals, and we asked the question, how does the constituent of the serums that we know so far contribute to sterol efflux come together? We took the serum HDL, which is the ApoB depleted serum HDL, UC isolated LDL, Lp(a), HDL, and supplemented plasminogen, all matching concentrations of total serum, what would be in total serum, and conducted an experiment where we assessed ABCA1 mediated sterol efflux of total serum and all its constituents. And here is the data. Total serum is the strongest effluxer. Then comes serum HDL, so HDL and all the other proteins, including plasminogen. You see isolated HDL, LDL, and Lp(a), they all efflux, Lp(a) dismal, almost nothing, but much lesser extent than total serum or serum HDL, and plasminogen was comparable to isolated HDL. But then we started putting the puzzle back together, and no matter how we put it back together, we've seen that it amounts to always higher than total serum. That prompted us thinking that maybe there are some inhibitory interactions happening in total serum that impedes ABCA1 sterol efflux. And we know that Lp(a) inhibits plasminogen's fibrinolytic activity. The data exists since mid-80s. And we wondered whether Lp(a) could also inhibit plasminogen-mediated sterol efflux. So we isolated Lp(a) from high Lp(a) patients and treated plasminogen-mediated sterol efflux with increasing concentrations of Lp(a), or we treated with 17 Kringle apo(a), which is believed to be the active fraction of Lp(a), the main protein on Lp(a). And both apo(a), the protein, and lipoprotein a, the lipoprotein particle, at increasing concentrations inhibited plasminogen-mediated sterol efflux. So now we have a new biology in place. Lp(a) inhibits plasminogen-mediated sterol efflux. And does this axis play a role in CVD? So we cannot directly answer this question, but we know that Lp(a) is an independent risk factor in cardiovascular disease. So it's highly regulated by genetic disposition, and plasma levels in a population can vary from 1 to 1,000 milligrams per deciliter. Its distribution is skewed. Only, depending on ethnicity, only 20 to 30% of the population will have 30 to 50 milligrams per deciliter. That's when the risk separation starts to appear. And Lp(a) has been shown to have pro-atherogenic, pro-thrombotic functions. And it's been shown to inhibit plasminogen's anti-thrombotic functions. So now we identify a novel biology for Lp(a), which is to inhibit plasminogen sterol efflux capacity. And Lp(a), I urge everyone who's listening to know their plasma Lp(a) levels. It's highly genetically regulated. You can't do much to change it. There are ASO inhibitors coming up, and PCSK9 inhibitors do lower it some, maybe 20 to 30%. But it is an independent risk factor. It really doesn't matter whether you're a female or male, what age you are, what HDL cholesterol you have. It is an independent risk factor. And poorly understood risk factor. So what we did is we went to an Lp(a) cohort, and we divided the cohort into low and high Lp(a) to see if their total serum efflux capacity is lowered. And I know I'm running out of time. I'm eating post time. I apologize. I'm almost done. So as predicted, high Lp(a) patients have lower sterol efflux capacity. And there is a nice negative correlation between total serum efflux capacity and high Lp(a) levels. And we are really switching the attention from HDL to total serum sterol efflux capacity, and which I think captures better what the plaque needs to answer biologically. And then we showed that it's really the high Lp(a) levels in the serum that are responsible to impede plasminogen-mediated sterol efflux. We see a nice reduction in supplemented plasminogen-mediated sterol efflux. And Lp(a) comes in different Kringle isoforms. That is, again, genetically mediated. You can inherit 8 Kringle from one parent and 40 Kringle from another parent, which creates an infinite level of complexity. And when we screened a patient cohort, in our little screen, we identified 9 different Lp(a) isoforms, and we had enough Lp(a), isolated Lp(a) from 3 patients with 3 different isoforms. And as you can see, it doesn't matter what the isoform is, all Lp(a) inhibits plasminogen-mediated sterol efflux. So we put on the table a new sterol acceptor that is highly present in plaque environment. Lp(a) is an independent risk factor that inhibits plasminogen-mediated sterol efflux. So in the evidence pyramid, we did a lot of work in vitro side of things. Animal research is very tough because mice don't have Lp(a), and Sam Simikas at UCSD have a mouse model with a definite Kringle repeats that really doesn't reproduce the human variability. We did quite a bit of work in human studies. Next, we are approved to carry this approach in MESA. And as soon as we get our grant, we will dive into MESA to see if total serum efflux capacity correlates with CVD risk, with atherosclerotic burden, and whether we can measure plasma Lp(a) levels, plasminogen levels. Can we come up with a coefficient that defines sterol efflux levels and build it in the risk prediction formula? That's the goal. So here are my collaborators and my beautiful environment at OHSU that allows me to go from bench to bedside almost seamlessly. And I leave you with this. Thank you for your patience. I know I ran over time. I thought I wouldn't, but I talked too much, I guess. So thank you. And I'm ready for your questions.
Natalie, that was wonderful. I am going to remind everyone who wants to ask questions to raise their hands. There is a blue button in participants that will allow you to do that. So yes, Z?
Okay. Can you hear me now?
Yes, we can.
Okay. So hi, Natalie. This is Z from Columbia University, and thank you very much for your wonderful talk. I'm sorry, my background, my daughter is laughing for some reason. So you showed a very interesting data that you at UPA and then showed it can lower the efflux, if I remember correctly. I'm just wondering that if you also tried the TPA as well.
We can cleave plasminogen to plasmin with TPA, and I haven't presented here, but we've seen the same results with TPA. TPA alone doesn't cause efflux, just as UPA, but when there is not enough plasminogen, because it's converted to plasmin to elicit efflux, we do see the same decrease.
Okay. That's very cool. And also, if I also remember correctly, both TPA and UPA, they also have 2 Kringle domains. So is there any interaction that you could predict with the ABC one?
So that's a very good question. So the TPA and UPA cleavage experiments are done such that we incubate plasminogen with UPA and TPA, pre-incubate. Maybe a competitive inhibition type approach might be more appropriate, where we co-treat the cells at the same time to see if the Kringles of TPA get in the way.
Okay, sounds very cool. Thank you.
Thank you.
I will send you an email. Thank you.
Thank you, Zay. One more question from Wen Dai.
Hi, hello.
Hi, Wen.
Hi, very nice talk. Thank you, Natalie. I have 2 questions. So the first question is that do you do some experiment showing that the plasminogen combines the lipids? I mean, some in vitro work to demonstrate. And the second question is that in the blood, in the circulation, would the plasminogen bind to some lipoproteins, just like ApoA do? Because we know that there is some very some common as for the structure between the ApoA and the plasminogen. So that's the second question. Thank you.
Then those are very important questions. And here, okay. So we did some, is my screen still shared?
Yes.
Okay. So we did some turbidity assays with the MPC, PUPC, and NBD cholesterol. And here is the data. So these are molar equivalent comparisons between plasminogen and ApoA1. So in a test tube with the regular turbidity assay, plasminogen seems to bind lipids as well as ApoA1. Okay.
Yeah.
So that's yes. Your second question is, what does plasminogen do with these lipids, right? Does plasminogen do full reverse cholesterol transport? I don't think so. But what plasminogen does, and we see it most of the time associated with HDL when we do HDL proteomics, not all the time, but most of the time plasminogen will be on HDL. I think what it does is it facilitates, it's like a shovel. It will grab and put it on the closest, biggest lipoprotein around. We see it on LDL as well, not all the time, but sometimes we see it on LDL as well. I think it acts like a shovel. At least that's how I think about it. I have no proof for it.
Okay, thank you.
So we will now move to Paul, if you don't mind, Natalie.
Yes, absolutely.
And I would love it if you can introduce Paul, because you know his career much, much better than me.
Yes, so Paul Mueller joined OHSU right after his PhD at UKentucky with Susan Smith in 2016. He's been working with Sergio Fazio, which he will present some of the work he did with him. He joined my lab about a year ago because he's growing to be an epigenetic scientist, and he wanted more exposure to omic big data research. And he's done wonderful work with me, but that's not what he's going to talk to you about. He's going to be talking what he's done with Sergio and tell you about his vision for what needs to be done from his perspective to address regression, atherosclerotic regression. Take it away, Paul.
So Natalie, you might have to unshare your screen.
Yeah.
Thank you so much.
Yep, cool. Thanks everybody for hanging out to listen to me talk. As Natalie mentioned, I've been working with Sergio, studying atherosclerosis regression, which I'll get into. And then since joining Natalie's lab, I've been able to add a little bit of omics and big data expertise to my repertoire, which I'll get into midway through the talk. This is a group that I probably don't need to reiterate the burden of cardiovascular disease, but I'll kind of emphasize the light at the end of the tunnel, and that is the the addition of the PCSK9 inhibitor to clinical treatment of patients with high cholesterol. And so I'm going to show you some data here from the GLAGOV trial, which was a see the
Is there a problem with Paul's connection?
Yeah, I couldn't hear him either.
Paul, there is a problem with your connection, it seems. We can't hear you. It's
Okay.
just, okay, now we can, there was like pausing. Okay. So we just lost him. We'll wait for a little bit until he comes back, and sorry about that. Sometimes connections are problematic. Okay, he is.
Hey.
Paul, okay, wonderful. All right, let's continue.
Sorry about that.
So can you screen share again, Paul? Oh no, you have a problem with your connection, it seems.
Internet.
Paul, you have an internet connection problem. All right. So,
you guys still can't see me. I can.
Okay. Let's just wait for another minute. Yeah. Okay. No, so I don't think we can connect Paul. He lost his connection completely.
Okay. So you guys can't see me at all then? Well, crap.
Okay, now we can see you.
Okay.
Let's give it one more shot.
Okay. Sorry about that, you guys. Are you able to see the screen share?
Yes.
Yes. Okay. So I don't know how much you guys heard from this one, but the bottom line was that PCSK9 inhibitor therapy was able to reduce mean LDL cholesterol to as low as 36.6 mg per deciliter.
Well, we lost him again. Paul, unfortunately, your connection, it disconnects and there's a lot of pause. So, and it's not getting fixed. So I suggest, okay, try speaking again. And if it doesn't work this time, we can postpone your talk to another date.
Or maybe we can try, maybe he can leave and then join again. That may also work.
Have you
Okay.
done that?
I'll do that.
Okay, let's see that. All right, let's wait for him to come back. Here is Paul again. Oh, that's a good idea. A suggestion to connect through the cell phone. Cool. Paul, if this doesn't work, you can try connecting through your audio or cell phone. Okay,
okay.
Let's try again.
Okay, sorry about that. All right. Okay, so blah, blah, blah. Aggressive statin therapy with evolocumab, the PCSK9 inhibitor, was able to induce atherosclerosis regression in about 2/3 of the patients, whereas another patients that saw regression, the plaque or the percent in atheroma volume was only reduced by about 1%, which is pretty modest to say the least, and probably suggests that plaque regression in humans is a slow process. But to give a little bit of hope here, the complete regression of atherosclerotic lesions is potentially feasible, although considered very rare. This is a case report published in the European Heart Journal earlier this year, where a 44-year-old woman with 70% stenosis in her left or in her LAD actually saw a complete regression of the artery following 3 years of just aggressive statin therapy. To talk about regression or the progression and regression of atherosclerosis, we like to think of progression in that influx of material, this cell disease, LDL cholesterol, and then followed up with our immune cells, as well as proliferating smooth muscle cells. And then following extreme lipid lowering or cholesterol management, you can experience regression, which we can think of as a net efflux of material from the artery wall, which includes reverse cholesterol transport and macrophage egress as well. Experimentally, several models of atherosclerosis, experimental atherosclerosis regression have been generated, including the aortic transplant model by Ed Fisher's group. This is a model where you take an atherosclerotic-ridden aortic arch and surgically transplant it into a wild-type mouse. This because of the low cholesterol levels induces rapid regression of the plaques. There's the reverse mouse model, wherein you can inactivate MTTP in an LDLR knockout mouse to correct hyperlipidemia. And then finally, the reconstitution of ApoE into ApoE knockout mouse. This has been done by Randolph's group with adenoviral delivery, and this is a model that we use with a bone marrow transplant as opposed to adenoviral delivery. So Sergio's group, before I arrived, had focused on the role of macrophage low-density lipoprotein receptor related protein 1. This is a backup receptor to the LDLR receptor traditionally found in the liver, but this is a ubiquitously expressed protein, and its knockout on macrophages actually accelerates atherosclerosis. And this is due to an increase in systemic inflammation, presence of elevated Ly6E high monocytes. There's increased TNF alpha in plaques. There's higher susceptibility to apoptosis in macrophages without LRP1. And as LRP1 is a doses see a decrease in efferocytosis, contributing to myocytes donors for all.
So unfortunately, we lost Paul again. Are you back?
Hey,
Yeah.
doesn't seem to be working, does it?
No, unfortunately. So have you tried calling from your
Yeah.
cell phone, Paul? That would be the last thing we can try. Like calling in from cell phone to connect.
I can. Okay, yeah, I can try that.
And if that doesn't work, we'll do another round with you after this. Mm-hmm. Okay, don't want to give up. I want to give Paul another chance. If this doesn't work, we're going to postpone it and do it another time. Okay, one last try, Paul. Go ahead.
Okay. Let's see. I'll turn off this.
Right. Yeah.